This video covers the different types of gene mutations. To start off, we're going to define some general terms and then we'll go into the specific types of mutations. So first, when we talk about a gene, we are talking about a section of a chromosome that codes for a trait, which really means it's the area that codes for a specific protein. And we talked about the specifics of protein synthesis last time. When we use the term gene mutation, what we mean is that there is a change or error in the DNA sequence. And these mutations can be helpful or harmful. So harmful mutation, when we think about, excess UV radiation or being out in the sun without sunscreen on, and that causes an error that can eventually lead to tumor formation or something like that, that would be an example of a harmful mutation. But sometimes they are good mutations that end up helping that organism be able to survive. So one example there would be, out in the desert, there's a volcano that happens, so there's sand, and then there's that dark, cooling rock there. Maybe an organism that lives there, like a mouse, gets a mutation that gives them a darker fur color so that they can hide on that black rock that's there. So that, that would be a helpful mutation. Because I know we think mutation, we automatically think bad, but they are not all bad. Sometimes they do help that organism be better at surviving and reproducing and passing those genes on. There are two types of gene mutations. The first one is called a point mutation, and the second one is called a frame shift mutation. So we'll talk about point mutations first and then move on to the frame shift mutation. The one type of point mutation is called a substitution. And in this case, one nucleotide is changed for another. So we're just swapping out one nucleotide. So maybe an example here would be that our original DNA reads T, A, C, A, T, G. There is a mutation that happens, and now it reads T, A, C, C, T, G. So we're just swapping out one nucleotide for another. And these mutations, we can classify them as silent, missense, or nonsense. So we'll do a description and the effect on those. With a silent mutation, the mutated codon codes for the same amino acid, which gives us no effect. So think back to our codon chart. We know that there are only 20 amino acids and 64 combinations. And for some of those, like proline, for example, there were 4 different codons that can give us that amino acid. So if there is a change, but it still gives us the same amino acid, we call that a silent substitution because it gives us no effect. It's the same amino acid, so our protein is still going to be the same. With a missense mutation, the mutated codon codes for a different amino acid. And the effect of that is going to vary depending on which amino acid it's gotten switched from. So let me backtrack for a second. Remember that if the DNA changes, mRNA will reflect that change, and then we're skipping ahead to what the amino acid will be here. Just don't forget that. A nonsense mutation happens when the mutated codon is a premature stop. So instead, it doesn't code for the same amino acid. It doesn't just switch amino acid. It goes directly to stop. And the effect of that is usually serious. What if our protein should be 150 amino acids long and the stop is right near the beginning? So maybe it's at amino acid, right after amino acid 25. If that protein, isn't complete and isn't made right, it will not be able to do its job. So the effect of that is going to depend on, what protein is it? Is it something that's necessary? Is it something we can treat? Things like that. So these are all different ways we can describe the substitution. Last, we'll move on to the types of frame shift mutations. Frame shift mutations give us a change in the reading frame. We know that mRNA is read in groups of 3. So if something is inserted or added or something is taken away, our groups of 3 aren't going to quite be the same. So there are 2 types of those. In insertion, where a nucleotide, oh my goodness gracious, what is happening to me? Okay, so insertion, a nucleotide is added. So an example, maybe our DNA is supposed to read TACGCA, and there is an insertion, so then it reads TACTGCA. So we've added in a thymine here. With a deletion, a nucleotide is removed or taken away. So maybe here our DNA should read TTACGA, and we have a deletion, and then it reads TTCGA. So that's going to change our group of 3s that are read. And we'll look at more specific examples of these and how serious the effects can be when we meet in class.